For me, it's down on the bottom. Perfect.
Okay. Well, hello everyone. My name is Maya Ibelaidene. I will be presenting research I conducted under Dr. Prue Talbot in the Department of Molecular Cell and Systems Biology. My presentation is a PRISMA systematic literature review on the effects of electronic cigarettes on early embryonic development. So this is the progress in which I will demonstrate the research I conducted over the past 2 years. Okay, so let's start with an introduction. So just some background information. Electronic cigarettes, more socially referred to as vapes, have become extremely popular, especially in the past decade, among people of reproductive age. And my research is concerned with the fact that pregnant women, usually pregnant women or women who usually smoke tobacco cigarettes, have either switched to smoking vapes or electronic cigarettes because of perceptions that they're safer than tobacco cigarettes. And some pregnant women have even been their doctors have even recommended that they switch to vapes while pregnant. And these vapes, that when people vape electronic cigarettes, they usually vape these with flavor chemicals. And so the purpose of my paper and my literature review was to determine if electronic cigarettes can actually harm embryos in pregnant women. So I'm going to walk through the methodology of this review. So I conducted a PRISMA search strategy, which is basically using Google Scholar and PubMed. I had a list of keywords or phrases that I used to collect as much literature as I could on this topic, ranging from the year 2000 until 2021. So my inclusion and exclusion criteria regarding this research was that I included papers that discussed the effects of flavored chemicals on embryonic development. And I also included papers that discussed the mechanisms in which these effects occurred. I excluded any papers that didn't talk about embryonic development because that's my focus. And I also didn't include papers that did not discuss flavor chemicals because those are extremely popular among electronic cigarette users. So papers that did not research that were not included in the review. Okay, so this PRISMA flow chart is basically a chart that demonstrates the papers that I collected. And so as you can see at the top here, I started with PubMed and a Google Scholar general search. PubMed and the general search yielded 352 citations. And after I removed duplicate papers, I was left with 327. After those, I looked at the abstracts for all of those papers and I excluded or included different papers. And at the end of that, I was left with 111 articles. And with those articles, I did a deep read and I again applied the inclusion and exclusion criteria that I mentioned. And so at the end of all of this, I was left with 40 papers from the year 2000 to 2021 on this topic. So let's discuss some of the results from this paper. So the results that I had, I split into animal studies and human studies. And so for the animal studies, the species that were observed in these studies were mice and Xenopus laevis. And the exposure methods meaning the methods in which the electronic cigarette fluids or airs were put onto the embryos or exposed to the mothers, was either through full body exposure, inhalation exposure, or just aerosol exposure overall. And there were a multitude of flavors tested in all of these papers, ranging from cinnamaldehyde or cinnamon flavor to citrus. The human studies focused on stem cell studies and researches. So the species or the cell types that were looked at were human embryonic stem cells, human pulmonary fibroblast cells, and HTR8S venial cells. And so the exposure methods, because these were cells and not actual subjects, they were exposed through doses of exposure ranging from 0.001% to 1%. And the flavors again tested ranged across the board. Less flavors than in the animal studies, but still a lot of flavors to look at. So in general, the results that came up, I'm going to start with the animal studies. So for the animal studies in mice, they found that mice that were exposed to electronic cigarettes had decreased litter sizes, and their pups had lower birth weights and birth sizes compared to their control groups. In addition to this, on the Xenopus laevis tests, they found oral facial defects, and these oral facial defects, a subsequent paper later on found that these defects were actually interacting with a pathway called the retinoic signaling pathway, retinoic acid signaling pathway. And so they found that electronic cigarettes are interacting or interfering with that pathway that's causing the oral facial defects in the Xenopus laevis. In addition to this, in a study on mice, electronic cigarette exposure caused inflammation in the lungs of both the mothers and the pups. So there's an effect going on in both the mothers that were exposed and the pups that were born as a result of exposure. Next, there were immune dysregulation in the mice as well, both in the mothers and the pups that were exposed to electronic cigarettes. And finally, again, with the mice studies, they found that pups exposed to electronic cigarettes and flavor chemicals during development had affected their brain development. So when it affected their brain development, that means that the pups that were exposed had interruptions in their learning and memory, and their learning and memory were severely impaired compared to the control groups. And in addition to this, they actually found neuroinflammation in the pups of exposed mothers compared to control groups. Now in the human studies as well, we see decreased birth weight and size, which is a significant indicator. And we also see on all the cell types that were studied, there were the survival of all those cell types were decreased as a result of exposure to electronic cigarettes. So now I want to talk about TRP channels, or transient receptor potential channels. The reason this is relevant to our discussion is because, as we'll find out, TRP channels are a diverse family of ion channels that can sense intracellular calcium and magnesium concentration. So TRP channels have been found to be extremely important in embryonic development. And as you can see in this image as well, a lot of these TRP channels, all of the ones listed here, are present in early embryonic development, and they all seem to be interacting with some kind of flavor chemical. So the reason this is important in our discussion is because what I'm going to explain as a result of this literature review, what I'm going to propose as a possible explanation for these effects is that the concentrations of these flavor chemicals could possibly be interacting with these TRP channels and causing the effects that we saw in the literature review. So now I'm gonna lay out my fetal exposure model that I created as a result of the literature review. So the first thing that's important to understand is that when we look at the pathway that oxygen or any inhaled substance goes through in a pregnant woman, for the most part, it goes almost directly to the fetus or the embryo. And in a paper from Khachaturian et al. In 2022, we learned that whatever people are inhaling from electronic cigarettes, they're retaining 100% of whatever they're inhaling into their lungs. And so when we know that information, this is a very important information because that tells us that essentially whatever is being vaped is being retained in the mother's lungs. And that's important because now we can, using that information, using that understanding, we can actually create an estimate of the concentrations that are reaching an embryo as a result of electronic cigarette vaping. So how this occurred, how this is created, is that we can take the concentration of a flavor chemical and divide that by the volume of blood in a mother. The reason we're dividing that by the volume of blood in the mother is that although everything being inhaled is essentially going directly to a fetus or an embryo, there is possibility of dilution as it travels to the embryo. So taking that into account, if we assume, for example, that a mother inhales 1 milliliter of a flavored concentration of a flavor chemical from an electronic cigarette, for example, if the concentration of a flavor is 155 milligrams per milliliter, we can divide that by 5,000 milliliters, which is approximately how much blood is in a pregnant woman. And we can come up with a concentration that is possibly reaching the embryo every milliliter. And so it's important to note that this is an estimation and there haven't been concrete studies on this. So this is just an estimation onto the possibility of concentrations reaching embryos. So when we look at this fetal exposure model, we can tell the example in the last slide was actually cinnamaldehyde. And so something important to note about cinnamaldehyde is that it's been featured in many papers as one of the most popular flavor chemicals tested in electronic cigarettes. And as you can see here, it's one of the highest in terms of concentration out of other popular flavors. And so this is important because as a popular flavor, it tends to be present in high concentrations in electronic cigarettes. So now that we laid out the measurement of this model, we can look at this. So as we measured, if, for example, if a woman is vaping 1 milliliter, we have our concentration here, and we can compare the values after the dilution to something called an IC50 value, which is something that just measures when a cell can detect a flavor chemical or anything really. And so that IC50 value in the literature for cinnamaldehyde on human embryonic stem cells was 0.00004 milligrams per milliliter. And if a pregnant woman inhales 1 milliliter, then the value after dilution is 0.031. And so that is significant. That's high compared to the IC50 value. And so what that means is that because it's larger than the IC50 value, then there is most likely going to be an effect on that kind of cell and on that kind of development. And so we will most likely see an effect based off of this estimated concentration and the IC50 value. And that we see that as well for vanillin. And menthol and nicotine also they're close in terms of IC50 value and the value after dilution. But what's important to note here is that 1 milliliter is not a lot. It's actually very little in terms of vape and electronic cigarettes. And so if we, for example, instead estimate that a pregnant woman vapes 10 milliliters, which is more accurate than 1 milliliter, or more probable, if we take these same numbers, these values after dilution are all increasing. And so that's increasing the effects that all of these flavor chemicals, these concentrations will potentially have an effect on the embryo during embryonic development. And so how does this relate to tryp channels? Well, I'm basically suggesting or interpreting this to be that if these concentrations are reaching these levels and they're higher than IC50 values, then a potential explanation for all the effects that we found in the literature review is that these concentrations are all high enough to trigger tryp channels and therefore affect embryonic development and be the explanation behind the effects found in the literature review. And so just a conclusion. So the use of electronic cigarettes during pregnancy in the literature review has showed that it does cause developmental and congenital defects in both animal and human models. And the exposure model supports the idea that these flavor chemicals and nicotine in the electronic cigarettes are reaching embryos at a concentration that's potentially high enough to trigger tryp receptors and explain the effects. And then these activations could potentially, as I mentioned, lead to the effects that we've been seeing in the literature review. So why does any of this matter? So these data show that electronic cigarettes does have a negative effect on animal and human offspring. And so pregnant women have often either been encouraged to or have done so on their own accord, using electronic cigarettes either as a replacement of tobacco cigarettes or other harmful substances because of perceptions that they're safer. And so this review will clearly show that pregnant women should not use electronic cigarettes until their effects on pregnancy are more fully understood. So thank you all for your time and your attention. I want to thank Dr. Prue Talbot for her mentorship and creation and completion of this project, and also Shabnam Enamadi for her contribution to this project as well. Thank you.